Hello, everyone, and welcome to today's webinar titled Development of Luminex-based Serological Test for COVID-19 Diagnosis and Monitoring. This webinar is part of an ongoing coronavirus virtual webinar series. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex. For more information about Luminex, go to luminexcorp.com. So let's get started. I want to remind everyone that this event is interactive. To participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of your presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Dr. Imad Tarhoni, a Postdoctoral Fellow at Rush University Medical Center at Chicago. For a complete biography of our speaker, please visit our Biography tab at the top of your screen. Dr. Tarhoni, you may now begin your presentation. Welcome, sir.
Hello, Susie. Hello, everyone. Good morning. I would like to thank you for this nice introduction, and I would like also to thank everyone that is attending this seminar today. In today's presentation, I would like to talk about our novel COVID test, the novel test for COVID-19 diagnosis and monitoring that we have been developing since the beginning of this pandemic. Before I begin, I would like to introduce my team. So beside me, Christina, Dr. Jeffrey Borgia, and David have been working tirelessly side by side to develop, optimize, and validate this test. And just one thing to mention here, because the patent of this test is pending, I'm a little bit limited on the amount of data that I can share in terms of test specifics. But I would be happy to share the validation results and clinical results. So the layout of this talk will be started with, we'll begin with a brief focus introduction on the COVID-19. Then I will describe the current strategies and testing methods for the COVID-19, followed by a description of the basics and the rationale for our tests, and then the validation results. Finally, I would be happy to take your questions and answer them after I talk a little bit about the future directives. And so let's begin. So coronaviruses have been known historically for being responsible for a couple of major pandemics on the world, including the SARS in 2002 and the MERS 2012. This one is considered to be a third hit by this fellow creature. In late 2019, basically in December, coronavirus have been identified to be responsible for a cluster of deadly respiratory distress syndromes in Wuhan, China. It rapidly spread, resulting in local epidemics and followed by global pandemics, which was announced by WHO in mid-March. In February 2020, WHO designated the COVID-19 for the disease, and designated the term SARS-CoV-2 for the virus. But for the convenience of this talk, I would be referring to a disease to the COVID-19, as a disease, and for the SARS-CoV-2 as virus. So let's take a closer look on the coronavirus. Coronaviruses are called coronaviruses. They are giving this name because of the shape of their the shape that they take because of those spikes on their surface. These spikes that are colored red here, they are colored red for illustration, but they are formed of glycoproteins that is responsible for binding to the angiotensin converting enzyme 2 receptor on the respiratory epithelial cells, and they facilitate the invasion of the virus. When we look at the deeper structure of this virus, beside the spike proteins, there are the envelope proteins, which work with the membrane protein to give that spheroidal shape of the virus and form the viral envelope. All that encloses the nucleocapsid protein, which also interacts with the genetic component, the gene of the RNA, the gene of the virus, which is the RNA, to form the nucleocapsid. I would come back to this structure because it's essential for the composition of our essay. So we'll go into details later. When we look at the pandemics figures, this slide, by the way, was created in June 13th. So far, the virus has infected more than 8 million globally, and it caused it took a life of more than 444,000 all over the world. Fortunately, half of this number half of these cases have already recovered. In the U.S. itself, there is a fourth of this number in terms of cases, and the fourth of this number in terms of deaths. The recovery is still low, which is about 25%. So when we look on more details, the U.S. is still leading the figures of the pandemics in terms of deaths and number of cases. So when you look at the death and the case fatality ratio, U.S. is still on the forefront of this pandemic. Brazil is climbing up now. In the U.S., the average number of cases detected daily is 22,000 per day. 66% of them are among those age group between 18 to 64. I would also come back to this age group because it's essential for designing the mass screening strategy for the COVID-19. But the reason why these numbers are high in the U.S. could be, according to CDC, could be due to the high testing capacity the United States is trying to implement. So far, in the U.S., we have conducted more than 23 million tests that can be averaged to 500,000 tests per day since the beginning of March, with the positive rates of 11%. So when the interesting thing is that how the trend is different among different regions of the world, and even as well as within the United States. So if you look at the graph on the left, the U.S. seems to be past the peak of this, hopefully past the peak of this pandemic. And now it's still showing fluctuation of number of cases. When Brazil, it's just started to climb up the curve of the cases. When you look at the right figure where we show different states, it is not the same for all the states here in the U.S. So as some areas are already recovered, like New York or New Jersey, there are some other states which are just started to climb up, like Arizona and Arkansas and Michigan as well. So we see some regions are started to plan for reopening and ease the restrictions, and some regions are just started to climb up the peak of the cases. That could be associated with with the nature of the asymptomatic nature of some of these cases, as we know that from 5% to 80% of the people tested positive of the virus have been they showed no symptoms or like mild symptoms in the past. Therefore, a large scale community level testing is critical to control the disease and reopen safely. So far, since the beginning of the pandemics, nucleic acid detection based on PCR has been the major laboratory test for diagnosis. But we have to mention that it's very specific, but not as good as we expect in terms of sensitivity. It's very specific because it detects the viral gene sequence, the RNA sequence that is specifically unique to this virus. But there are a lot of technical issues that I will describe in the next two slides regarding the sensitivity that causes the low sensitivity of the test. For that reason, FDA announced the emergency use authorization guide in February 2002, describing the policy regarding the immediate use of tests developed by certain certified laboratories. The objective of this guide is to increase the testing capacity in the United States. So what are the current testing methods for the COVID-19? So beside the PCR, the reverse transcription quantitative PCR, which is depending on detection of a nucleic acid in the swab from the nasopharyngeal or oropharyngeal swab. We have other tests like detecting the viral antigen in the same sample. As far as I know, there is only one test that is approved by FDA and for which we don't know that much information in terms of validation. Currently, the very common test that used in large scale is the serological immunoassays. These immunoassays or serological tests are planned to detect the immunized patients or patients who developed immunity to the disease, to the virus. Basically, they measure the immunoglobulin M and G. So nowadays they are widely used in lieu of the PCR. The rationale for that is that PCR has a couple of limitations. So PCR requires high-quality nasopharyngeal swab it should contain a sufficient amount of viral RNA. To do this, you have to successfully follow specific techniques that requires certain training by operators and requires successful transport of media and extraction steps. And in addition to that, the amount of viral RNA may vary from within the same patient and according to the different times when you take the test, and also this procedure can be unpleasant to the patient, and sampling techniques can depending on the operator, as I mentioned. All these factors can lead eventually to insufficient amount of viral RNA that lead to a false negative result. In addition to that, as I mentioned, this procedure requires high trained personnel from the step of acquiring the sample to the step of producing the results. For that, this can be done in lower scale, like in daily routines for hundreds of cases. But when we talk about large groups in the pandemics, that means you're trying to test thousands of cases per day. It's going to be overwhelming to the medical facilities, and there will be a huge backlog of tests and results. The turnaround will be very high, I assume. So the rationale for using the immunoassay tests in lieu of PCR in terms of screening it can be described in this slide and this figure. So if we look at at the beginning of this graph, in day 0, that indicates the onset of symptoms. If you look at the evidence of the virus, you will see the virus on the samples. So either the viral RNA or the antigen, they will be high in the biospecimens, either the nasopharyngeal swab or the urine or the blood. They will be high. But that will go down by the time. And according to average of the time that was reported, it goes down by the day 6. And these numbers are just averages. In terms of immunity, we see the immunoglobulin M, that's the red line, and the immunoglobulin G, they both start going up or responding in average of 3 to 4 days. So what I'm trying to say here is that if you test for the RNA in the first 3 to 4 days of the onset of symptoms, you will see high sensitivity of the test, because you will detect the RNA, assuming that you perform, as I mentioned, you perform an optimal procedure. And if you test beyond the day 6 or day 5, you most likely to see a lot of false negative if you run the PCR. Compared to the immune, when we look at the immune detection tests, it will be also it will yield high false negative at the beginning of the symptoms because there is no immunity, but it will be highly sensitive and at high sensitivity in the like beyond the day 6. So let's talk about because this, based on this criteria and the difference between the two tests, the performance of those two tests, CDC announced the surveillance of U.S. serology testing strategy. With this, the objectives of this strategy is to provide more complete estimate on how common the virus or the disease is, and to guide the the control measures such as social distancing and reopening. The question CDC is trying to answer using this method or this strategy is that how much of the U.S. population have been infected without showing symptoms, and how much of the U.S. population have been infected generally. And if these antibodies or this immunity can be associated with the protection and from future reinfection, and if they have any therapeutic implications. So let's describe the current methods of serology testing. So the majority of these tests are based on the lateral flow assay techniques, which is described in this slide. So basically, this is the card that is simply you load the sample on this card, and then you load the buffer. The samples will flow laterally through different phases. The first phase is attached. It contains gold COVID-19 antigen conjugates. And once these antigen conjugates encounter anti-COVID-19 antibody in the sample, it will show this light red line on the step 5. And then you also have a control, which is a rabbit gold conjugate, a control indicator. So as the sample goes forward to the second line, which is C here, it should light up to show that the control is positive. So you know that the test is valid. So in step 7, you see that the positive test should contain the line for T, the test, and line for C, the control. And the negative test should contain only the line of C, that is the test is valid, but no line for T. And if there's no C line, the control line, then the test is not valid anymore. So There are, so far, there are tens of approved FDA serology tests. I'm just showing a couple of them now. They are different. There are different vendors. There are different manufacturers. Abbott, Celix. Abbott and Celix are the most commonly used, but you see here they have different performance, from sensitivity of 100% and 93%, even though they are measuring the same target, the IgG for nucleocapsid or the IgG for spike. One thing here is that I didn't show it here in this slide. The majority of these tests have validated this performance after day 12 of the onset of symptoms. And some of them showed these figures of sensitivity and specificity after day 14. So now let's talk about the rationale or the basics of our Luminex bead-based tests. So our test is based on the platform invented by Luminex XMAP bead-based technology. Basically, this technology is based on the reaction or conjugating the antigen or the target on the paramagnetic microsphere or beads. These beads are internally dyed with two colors, the infrared and red fluorophores. These two colors would give the beads, they are in different proportions, so they give those beads different color region or different ID that can be detected and can be distinguished from the other beads. So you can you have more than 500 bead regions, and you can easily distinguish between those beads based on the color. So in order to develop this assay, basically you have the bead, and you can, using the carbamide based reaction, you conjugate the protein or the target or the capture molecule on the bead, and then you expose this. You expose this bead to the sample, serum, plasma, urine, or any kind of liquid biopsy sample. And then you add the secondary reporter that is conjugated with biotin, and then finally you add the fluorophore. So there are different formats of this assay components or this assay. This one specifically, in this one I'm describing what we did for this test. This is called indirect assay for antibodies, is where when you conjugate the antigen on the bead in order to detect the antibody on the serum. The other formats are if you play with the arrangement of this assay, you actually conjugate, if you actually conjugate the antibody on the beads, you can detect the cytokines or the protein that is soluble in the biospecimen. With that, we call it capture assay for cytokines. It could be cytokines, hormones, soluble receptors, ligands, any form of protein that is soluble. The third form of this assay would be specifically for the nucleic acid, is when you have tag and anti-tag react to each other and report it with the fluorophore. Finally, these beads will be mixed with each other. I mean, imagine you're performing a multiplexed assay where you take in each of these bead regions and conjugate it to a specific target. Let's say you have a bead 1 for IL-6 and bead 2 for TGF-beta. Now, when you mix them together, and you can, as I said, you can mix up to 500 beads, and now even more according to the next generation instruments developed by Luminex. Now, if you in the Luminex instrument, you expose these beads into two forms of laser beams. The green one is to detect the target. The red one is to detect the bead region. That's why the multiplexing capability of the instrument. So if the beads will flow via a stream, like in a streamline, like the flow cytometry technique, and both lights will be able to detect the bead number or the bead ID and the associated or the conjugated target, the amount of target as conjugated to these beads. One thing to mention that for illustrative purposes, I draw one molecule here, but you can imagine on the surface of these beads, you can conjugate as many as millions of molecules. So you can detect millions of target molecules on the surface of these beads. That's why it's considered highly sensitive compared to conventional methods like ELISA. One thing also to mention, one of the advantages of this is immobilizing the target on those freely mobile beads. It allows more interaction with the sample, with targets on the sample. That's why you see also high sensitivity of the results. So in order to design this assay, you have to go through different phases. So first, you have to identify your targets, and this is usually based on different methods. So either you go based on the bioinformatics analysis of currently available data to identify your targets that you're gonna be testing for. Second is, or the second option is to go based on the knowledge of the pathological pathways or the pathways responsible for the diseases, and you select your molecules of interest. A third way is to follow the analytical proteomics approach, like mass spectrometry and basic proteomics approach, to identify your potential candidate target, and then you design this assay. Second, you purchase the reagents. And this is not an easy process. Actually, you have to figure out which the best reagents that you can implement in this test, and you have to validate them to make sure that they work together. Then the third step is the coupling reaction to couple those capture molecules on the microspheres, and then finally you optimize the assay in terms of assay performance specifics, like the assay linearity, the dynamic range, and the cross-reactivity, and so on. So this is the form of our assay. So we have 4 bead regions. We conjugated those 4 main components of the coronavirus on each bead. So we designated one bead for the spike and nucleocapsid, and for the other bead for the nucleocapsid, and so on. So we have 4 unique bead regions, for one for each spike. One for each antigen, and we multiplex them together, and we optimize the assay according to the white box that you see on the left. So we optimized all these specifics: the linearity, detection limits, sensitivity, precision, cross-reactivity, and so on. We then validated the test on the 2 groups, the COVID positive group and the control group. And then we read them on the Luminex platform, and I will report the results later. So this is just a description of the assay. These are indirect immunobead assays. It contains the spike, nucleocapsid matrix, and the envelope proteins, or protein targets, sorry, to capture those antibodies specific for those. They have. We are looking for to improve this performance, but we already tested the IgG, IgA, IgM, and also the total IgM. So to start with, as I mentioned before, I have some limitation to or restriction to declare much of data here. But here I am showing an example of the assay range and the linearity of the spike and the nucleocapsid, for example. We go into, we were able to get sensitivity as low as decimals of picograms per ml. And we, again, one of the advantages that I didn't mention of this test is that we capture the 4 of them at once. So even if you even if the patient did not develop the antibody for spike, you're not going to miss it. If the patient developed antibodies for nucleocapsid, we still can test it positive based on the algorithm that we developed later. So the first validation we did is to accommodate the method of specimen collection, which is based on the dry blood spot. Basically, these cards are available for low costs, and it can be shipped at home, like shipped to the patient house, and we can perform the finger prick test and just send it back by USPS. Here we show in the difference. So let me talk about these figures. These are actually the first validation group is that it's the 10 positive cases and 10 control cases, and you can see the difference in the level of the MFI. You can clearly distinguish the positive from negative by the difference in at the level of the average MFI. Here in the light blue color or the turquoise color, it's the spike plasma concentration, spike antibody. And the light one was the one that it's measured from the dry blood spot. So you see it followed the same strange. That's why I'm showing this line graph, is to show that they follow the same pattern, but with a little bit lower intensity of the MFI. This can be optimized, but what we're trying to show here is that the feasibility or the applicability of dry blood spot to collect sample for this test. The second validation we try to test is the accommodation of the 38 4-well plates in layer of 96 well plates. So 96 well plates. This is how we performed this test so far. All the steps that we performed were on 96 well plates. With 96 well plates, you can perform 40 samples per plate. But with 384, you can test for 184 samples per plate. So with that, we try to show that we can accommodate this platform or this plat format, or in order to accommodate more testing capacity for this platform. And here in the figure, again, you see the turquoise color is based on the 96 well plates, and the intensity of the MFI went a little bit down, but it still followed the same pattern when we tested the samples on the 384 plates. So again, it's when you accommodate this test, you need to optimize it, but it's still showing the same results, the same sensitivity and the specificity performance. And then we also validated the timing. So we considered that shorter time will be helpful to allow this test to be more feasible in this pandemic strategy. So the regular time for the test is 8 hours total. But in order to scale it down to a couple of hours, we showed that first we can optimize the secondary antibody. We showed how can we optimize the primary antibody incubation from 4 hours to half an hour. You see here the white color is for 4 hours incubation, and the light blue color is for the 30 minutes incubation. Of course, there is a decrease in this intensity of the MFI, but it still shows the same pattern or the same sensitivity to distinguish the COVID-19 positive from the control. And with that, we can scale down the performance time of this assay to the half, that's 4 hours or 3 hours. We also did the same thing with the secondary. So we tested the 1 hour incubation versus 30 minutes incubation, and again, it showed not that much different in terms of the MFI signal. So we still can accommodate this this time too. So the total test will be around 4 hours or 3 hours. Finally, we did the validation with clinical samples from with the clinical samples from larger cohort. So we first selected the control cohort, around 262 cases from the collected during the peak of flu season. The peak flu season. That's collected from November to March last year in our biorepository. With that, we aim to make sure that this test does not cross-react or give false positive with other flu-like viruses. About 75% of those cases were COPD, that's chronic obstructive pulmonary diseases cases in this cohort. And they did not test positive with PCR for COVID-19. The positive cohort was 180 samples collected at Rush University Medical Center from March 1st to April 10th. And they all tested positive, and they showed symptoms of coronavirus, of COVID-19. And just one thing to mention about the drawback of this cohort is that it has been collected regardless the onset of symptoms. So we break down those cases according to the time when their samples were collected. So 128 samples were collected in the first 2 days. And I put this figure again to remind you that during those first 3 days, you expect to see low sensitivity of the serum test, of the antibody test. So as we expected when we run the test for the both control and positive group, we saw low sensitivity, and but I just have to mention here that when we run the test, we developed that classification algorithm on the using RPART package on our program to come up with a classification algorithm to help using those 4 targets to classify the patients or to predict the diagnosis. So here's the performance. The sensitivity in overall results, regardless the time of PCR test, the sensitivity was 55%. The specificity was 98%, and accuracy was 80%. And the negative predictive value was 76%. Now we optimized this cohort to include only people who were tested 3 days and beyond, 3 days and beyond the onset of symptoms. So we applied the same algorithm, the same classification algorithm, and we come up with better or improvement of the performance of the sensitivity and specificity. So the sensitivity was 92%, specificity was 98%, and accuracy was 97%. Then we went further to include only 6 days and beyond. And I want to remind you that the majority of the tests are under, according to the FDA, sorry, I didn't mention this, but according to the FDA recommendation is to test, to validate these tests is to test beyond day 12 of the onset. And now we are trying the day 6 and beyond, and implemented the same classification algorithm that we developed. And we ended up with sensitivity of 100%, specificity of 98%, and the accuracy of 98%. Now, we went to the second phase of the validation when we increased the control group to close to 1,000 patients. And with that, we aim to use, according to the recommendation from FDA, is to use as much large cohort as possible from the control to make sure that this test is applicable on the as a screening test. And the control group contains 44 patients collected in 12 days. So with that, we redesigned the classification algorithm using our program to show this, to end up with this classification system. So let me describe this classification algorithm briefly. So you start, when you have the sample, you look, the algorithm will be automatically run to first look at the spike antibody. If it's more than 40 picograms per ml, it will go automatically to look at the nucleocapsid antibody. If it is more than 24 picograms, then the patient is positive. If it's less than 24 picograms and also less than 40 picograms of spike, the blue colored is, then you look at the membrane antibody. If it is negative, that is less than 1 microgram per ml, then the patient is negative. If it is more than 1 microgram per ml, you look at the envelope antibody. If the envelope antibody is more than 28 micrograms, the case is positive. And if it's less than that, the case is negative. So in this, just one thing to mention that this is all bioinformatics system. So you basically run the raw data on the bioinformatics program, and it will come up with this decision, either positive or negative. So when we did this test on the 1,178 patients, we came up with sensitivity of 100%, specificity of 99.6%, and accuracy of 99.6%. That is outstanding performance comparing to other tests. And also, we are presenting a multiplexed method to test 4 targets at once, or 4 antibodies at once. So we make sure you do not miss this case, even if the patient did not develop antibody for one of them. So again, these are the performance of the other of the current tests, which is comparable to others. Now, when we talk about how can we implement this test, generally, if you want to incorporate it with PCR testing in order to diagnose or plan for back to work or reopening. So simply, if you have negative PCR test, that means there's no indication of the virus in the body. And if you have also a negative antibody test, that means you haven't encountered this virus before and you are susceptible. So the decision that can be taken based on that is to maintain social distancing and stay home. If the PCR was positive and the antibody was negative, that means you are infected, but you don't have immunity yet. You're supposed to go into quarantine. You are infectious, simply. If both tests were positive, you are also in infectious phase, and you have the virus and you have the immunity, so you are still in. You need to be in quarantine because you're still in infectious phase. However, when the immune test is positive and the PCR is negative, that means the patient has encountered the virus at some point in the past, and then he developed the immunity. This will make him or make that person categorized, or that we can categorize this person as a safe person, and he can go back to work, go back to school, and without being concerned to infect or spread infection to other people. So the immediate application of this test is to provide a convenient and cost-effective means to test non-vulnerable populations or non-symptomatic populations, mainly those groups that I mentioned before, the 66% young and middle-aged groups, who are, we expect that those are dynamic portion of the population. They go to schools, they go to work, they hang out all the time. You can screen those people on large scale and decide and help to plan to return to work or stay home decisions. With this test, we envision that we provide means to evaluate plasma in the blood banks for therapeutic, conventional therapeutic plasma to use based on the strength of immune response, to the COVID-19 virus. And we also can use this test as a companion diagnostics method for to monitor the vaccine efficacy and duration of antibody titers. It can also be convenient to epidemiological monitoring of infection rates worldwide, and as I mentioned before, according to across the regions of the countries or the states. It also will relieve the burden of the clinical laboratories struggling to keep up with the PCR-based tests of active cases. The backlog of these tests, now usually they run them in 3 days or 2 days. The future directives is regarding this test is that we're gonna refine the assay and transform it to automated workflow. And again, we're going to accommodate 384 well plates and optimize it further for dry blood spot method. And we are actively collecting more samples from Rush University Medical Center to prepare for FDA validation in the next 2 weeks. Using the emergency use authorization. We have multiple federal funding mechanisms being pursued for Chicago-based research networking, and we are open for collaborators. This is, like I mentioned, the Rush is doing an ongoing collection of samples and admitting patients and screening patients in the community in Chicago. So we are also collecting samples in our biorepository to further validate this test. To conclude the slides, I wish everyone to stay home and practice social distancing. And hope if you have any question, please ask me now in the QA session, or you can contact Dr. Jeffrey Borgia on his email shown on the slide. If you have any additional question or interest to collaborate in research or license agreement. Thank you so much, Susie.
And thank you, Dr. Tarhoni, for your informative presentation. And we will now start the Q&A portion of the webinar. So if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So Dr. Tarhoni, we already have several questions coming in from our audience members. And thank you all for joining us today. What's the plasma sample dilution in the slide shown?
So we did 1 to 500 dilutions. We tested multiple dilutions, but currently we identified 1 to 500 as the best.
Thank you for that. And the sensitivity is low, 55%, when samples within the first 3 days on symptom onset, and much better starting from 6 days. How early can we consider this test with acceptable accuracy?
So like I showed, we expect accuracy comparable to current tests at day 6. So, which is still an outstanding performance, because a lot of these figures that we saw from other tests are tested at day 12 and beyond, and we were able to show the same figure at day 6.
Thank you. And are there any other serological tests which only detect one target but have the clinical validation performance as yours? And if there were, why could they achieve it without using your multi-testing strategy?
Can you say the question again? Sorry.
Sure, of course. Are there any other serological tests which could only detect one target but have the same clinical validation performance as yours?
There are actually, what I showed, the current tests available, they all have the same, the majority of them have the same characteristics, but this is according to their validation studies. And what I'm trying to mention here is I'm not like criticizing anybody, but a lot of these tests, when they were tested from other institutions, they show different characteristics, different performance. And yeah, so there is a lot of debates. There are a lot of debates on the actual performance of these tests, but they are still like outperforming tests. That's how can I say.
Thank you for that. We have time for a few more questions. Can you collect plasma from a patient who still has positive results for the virus?
Yes, of course. Is that the plasma for diagnostic purposes or for the therapeutic purposes? Are we talking about?
Perhaps they can rewrite. They'll have to rewrite there in the question, and we can answer that in maybe written. And why do you choose a cohort of age median of over 60 for your clinical validation?
So this is the cohort in our biorepository. Second, these are the most vulnerable group for the COVID-19 tests. And as I mentioned, majority of these tests at Rush University were conducted on those patients who were admitted in Rush University Medical Center. So these have, those are the majority of them are in this age group. Speaking of the control group, these are the patients that we screened for lung cancer previously, and they have this average of average of age.
Thank you very much. And what is the dilution with dried blood spot?
Okay. The dilution, we're still optimizing it, but that dilution that we did at that time was 1 to 500 as well. We're testing different dilutions as of today. We're testing another dilutions for dry blood spot.
Thank you. And again, audience members, great questions coming in. Any questions we can't answer today will be answered via email. Dr. Tarhoni, is your test qualitative or quantitative?
It can be both. It's what we run now is quantitative test with a standard curve, and we can perform it on a qualitative basis as a positive, negative.
Thank you. And our final question, how did you validate the quantitative part of your algorithm? Do you have controls for this? And if so, what are they?
Yeah, that's why this is the initial quantitative test was based on the two things. So first was the recombinant proteins and the antibodies from other species. We also used the standard curve from serial diluted samples, known for being positive. But of course, we're now pursuing actual purification of the human antibodies to run the quantitative standard curve. We're looking for these. Yeah.
Dr. Tarhoni, thank you for your presentation today. Do you have any final closing remarks that you'd like to say to the audience before we close today?
No, actually, just I want to stress on the advantage of multiplexing strategy when you develop tests versus using uniplex or one target. We've been known for, as a group, to develop the multiplexing tests for screening of lung cancer and developing prognostic tests for lung cancer. And we encountered that we experienced the difference between looking at one target at a time versus looking at different biomarkers at once. It's more informative, more accurate, more sensitive, and more it outperforms other platforms.
Dr. Tarhoni, thank you for your presentation and for your time today and your important research. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Luminex, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand through the end of 2020. Labrets will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay safe, take care, and bye-bye.
Thank you. Thank you so much, Susan.